Miguel owes you 7 minutes. So the last speaker for this conference is Professor Gil Ast from Tel Aviv University, and he will talk to us about familial dysautonomia.
Thanks a lot. I'm really, really stunned to see you all. I was absolutely sure that this is going to be a talk between Miguel and I. He has a personal interest in what I'm going to say. I know that if I were you, I would long be sitting at home in front of the TV watching the US Open. Let's give it a try. Last 15 minutes, Ran, you gave a beautiful talk, so I'm going in sympathy with you, I'm going to cut my talk and to see the US Open. Okay, so I'm going to talk now. No. Poznan.
And give my talk again.
And Yossi Greenbaum will have the last 10 minutes to do whatever he wants. Okay. So, familial dysautonomia is a neurodegenerative disease that affects exclusively the Ashkenazic Jewish population. The mutation is in a gene called ICAP or BCAP, ICAP for sure. It's in the beginning of intron 20, causing exon 20 to shift from constitutive inclusion to alternative splicing, meaning in FD patients we see the two mRNA isoforms, with and without exon 20. From this isoform, there is production of the functional protein. From this one, no protein is synthesized at all. So this is a tissue-specific alternative splicing disease. So when we started this research, basically I really need to ask Miguel to thank Aaron Razin. Aaron is the one that forced me to get into it. I don't like to do anything that is related to diseases, but I'm really grateful that he forced me to get into it. So when we started this project, we generated 3 different cell lines from 3 different FD patients, 1 parent and normal cell lines, and we detected something amazing. There is a decrease, gradual decrease in the level of the full-length mRNA between wild-type, heterozygous, and the FD patients. Nevertheless, despite the fact that there is a 60% decrease in the full-length mRNA between the wild-type and the heterozygous produce the same amount of ICAP protein as the normal. But then the 50% reduction between the heterozygous and the FD patient totally shut down ICAP translation. This is not degradation of the mRNA or the protein. So it seems that if you can elevate a little bit the mRNA from this level to this level, you can restore protein production. So with that in mind, we decided to search for therapy. And we decided to search either for therapy that has already been approved for other neurodegenerative diseases or for drugs that do not require FDA approval. For example, food supplements that are known to affect the neuronal system. So, making a long story short, we decided to zoom in on phosphatidylserine. Phosphatidylserine is a compound that we produced in our body. It is located in membranes, mostly membranes of neurons. It is involved in synaptic communications and in neuronal apoptosis. During our lifetime, there is decrease in the amount of phosphatidylserine that we produced in our body, and it was approved to be labeled by the FDA that if you consume phosphatidylserine as a food supplement, it will elevate your long-term memory. So this is the link to the neuronal activity. And most importantly, you can consume high amount of phosphatidylserine without any side effects. So giving phosphatidylserine to the cell lines that we generated from FD patients, we see 3-fold increase in the level of transcription from the ICAP gene without changes of the splicing activity, and 2.5-fold increase in the synthesis of the ICAP protein. So phosphatidylserine elevate transcription from the ICAP gene without changing between exon inclusion and exon skipping, and it elevates synthesis of the ICAP protein. So how phosphatidylserine basically activate this transcription? To make a long story short, we demonstrated that phosphatidylserine bind to the membranes of the cells, activated the MAP kinase ERK pathway, leading to activation, phosphorylation of two transcription factor ELK1 and CREB that bind to the promoter region of ICAP and elevate transcription. This partially releases the FD cell lines from partial cell cycle delay, improving their mobility in a wound healing assay. So we found the mechanism by which phosphatidylserine elevate transcription from this gene. We then decided to generate an entire organism in order to examine the effect of phosphatidylserine. So we produced a humanized transgenic mice in which we replace in the ICAP gene exon 20 and the two flanking introns with a human sequence and with the FD mutation. These mice show shift in the splicing of exon 20 from constitutive inclusion to different level of alternative splicing in all tissues. But the level of exon skipping in the neuronal system is presumably not sufficient enough in order to inflict the human FD symptoms. But these mice are very powerful model organism in order to study the effect of drugs on transcription and splicing. So we supplied those mice with phosphatidylserine for 3 months, extracted different tissues, and demonstrated elevation in the full-length mRNA, the one that can produce the functional protein, by 2.5-fold, and elevation of 1.5-fold in different regions of the neuronal systems. So in internal tissues, we have a very nice elevation of the ICAP mRNA level, but a much moderate effect on the neuronal tissues, and I'm going to get back into it within a minute. What about the synthesis of the ICAP protein? 8-fold increase in the liver, and a minor effect, but still a very significant one on the neuronal tissues. We then perform microarray analysis, and we found on the cerebellum, and we found that 2,400 genes were affected. Remarkably, quite a lot of them are related to a different neurodegenerative disease called Parkinson. So it seems that Parkinson and familial dysautonomia share molecular mechanisms, and it might be that phosphatidylserine is going to be beneficial also for Parkinson. So at that stage, Horacio Kaufman, who is the physician from NYU that treat FD patients in the East Coast of the United States, took 7 patients, examined the level of the ICAP mRNA in their blood at time 0, supplemented them 3 months with 300 milligram of phosphatidylserine and another 3 months with 600 milligram of phosphatidylserine, and he detected a beautiful dose-dependent effect, elevation in the level of the full-length mRNA in their blood, indicating that phosphatidylserine is going to be beneficial for FD patients. So one of the cavities with phosphatidylserine is that this is an oily molecule that have a problem to cross the blood-brain barrier. This is the reason that we need to treat for a long period with phosphatidylserine in order to get the effect on the neuronal systems. So we decided to try to overcome this problem and to overcome the blood-brain barrier. So we developed 2 different assays, 2 different techniques. I'm going to talk only about the upper one. This is nasal admission. In collaboration with a company in Israel that is called Sypnose, they developed this aerosol system that releases phosphatidylserine with very high pressure as aerosol into the upper part of the nasal cavity, and from there by diffusion into the brain. So we fluorescently labeled phosphatidylserine, and after 2 hours of spreading of spraying the mice, you can see huge amount of phosphatidylserine all over the brain. 24 hours after that, you can see that the phosphatidylserine is channeled into the spinal cord and from there to the preferential nerve system, which is extremely important in FD. So we found a way in order to overcome the inability of phosphatidylserine to cross the blood-brain barrier by the nasal admission, and we hope that this is soon will get into the clinic. There is a clinical trial that is going to start in Sheba, and we hope that after 1 year that they will start with this nasal admission. Okay, so then we decided to ask what is the molecular mechanism that is impaired in these neurons. It was shown more than 30 years ago that a group of neurons that are called the dorsal root ganglia that are located along our back. These are the ganglia that collect all the signals from the internal tissues, sending them via the DRG, the dorsal root ganglia, into the spinal cord and the brain. These DRGs are much smaller in FD patients compared to normal individuals, and we decided to ask what is the missing molecular mechanism or the impaired molecular mechanism in these neurons. So everything that I'm going to show from here on was done in collaboration with Eran Perelson, who really took us into the neurodegeneration field. Okay, so we generated a conditional knockout mice in which we are removing exon 20 only from the DRG during embryo development. Removal of exon 20 from the DRG until embryonic day 10 is leading to embryonic lethality. In then 10 and then 10 and a half, removal of exon 20 is leading to viable mice that have most probably all of the human symptoms for FD. They are much smaller, they have gastric problem, ataxic problem, they have a much smaller DRG, their preferential nerve system are much thinner, and they form a totally different branching from normal neuronal systems, and growing the DRGs on a cover slips show totally different morphology than normal DRGs, and some of the axons are returning back on themselves and touching and forming synapses on the same axon, something which is a big problem in neuronal development. So we have a system in which we can ask what is the problem, molecular problem, with these DRGs. So in collaboration with Eran took the DRG from these mice, grow them on the cover slips, and he added labeled neuronal growth factor to the distal part of the DRGs. And he examined the movement of the labeled neuronal growth factor along the axons. And he found a much slower movement of this cargo along the axons compared to normal neurons. So the neuronal growth factors are moving along the axons on highway. They are cargo that are moving along the microtubulin the highway. So we decided to ask whether this highway is impaired, and indeed so. We detected a much lower level of acetylated alpha-tubulin. The microtubules are incorporated from acetylated alpha and beta tubulin. So there is a much lower level of acetylated alpha-tubulin and elevation of HDAC6. HDAC6 is the enzyme that is responsible for the removal of the acetylation from alpha-tubulin. So in these neurons, there is a problem with building the highways along the axons by the level of reduction of acetylation of alpha-tubulin and elevation of the enzyme that is responsible for removal of the alpha-tubulin. Can phosphatidylserine correct this problem? And the answer is yes. When we are treating different cells from FD patients with phosphatidylserine, we detected elevation in the level of ICAP, elevation in the level of acetylation alpha-tubulin, and reduction in the level of HDAC6. and better movement of the neuronal growth factors along those axons. So phosphatidylserine can definitely reach the target and can definitely improve what is impaired in these neurons. And indeed so. We supplemented the humanized FD mice for 3 months with phosphatidylserine, and we detected elevation in the level of ICAP in their neurons after this treatment. Okay, this is the final slide. The question that we are going to ask, can we use 2 drugs in order to have an additive effect, which means one of them gives X, the other one is give Y, and we can get with the 2 of them a much higher effect than each one alone, and long story short, the answer is yes. We are using phosphatidylserine that affect transcription only. But there was a work from Sue Slagenhoff from Harvard that found a different drug called kinetin that elevate splicing of exon 20. You can see here the 2 mRNA isoform in FD cell lines. When we supplemented those cell lines with kinetin, you can see a beautiful shift into exon 20 inclusion. However, there is a price to pay. Kinetin is toxic, especially in the recommended dosage. So we decided to go down with the level of kinetin to the minimal needed. Most probably it's not going to be toxic to the patients, and to see whether when we are adding phosphatidylserine, it will give us a higher effect. So just with phosphatidylserine, the elevation in the ICAP protein is 2.5-fold. With lower amount of kinetin, the elevation is 7-fold. Combining 2 of them together give us 8.1-fold, indicating that both of them operated on 2 different layers, combination of both of them giving us a synergistic effect. For those that do not know, my lab is basically an expert on the link between chromatin organization and splicing. And we demonstrated that changes in splicing cause changes in chromatin organization. So we decided to ask whether we can open the chromatin and affect splicing, and surely do. We use a HDAC inhibitor that is called TSA to open the chromatin, and this shifts splicing from this mode of splicing to mostly exon inclusion of exon 20. This leads to elevation of the ICAP protein by 3.5-fold. Phosphatidylserine alone, 3.5-fold, both of them 4.5-fold, indicating that they have a synergistic effect on splicing, and most probably to treat FD patients, you need to use multiple drugs in order to have a higher effect. So what I've shown here today is that we have identified a potential drug that can treat FD patients, that we found the molecular mechanism by which this drug is activating on the level of transcription. We found how to overcome the inability of phosphatidylserine to pass the blood-brain barrier by nasal admission. We found how what is impaired in the neurons that are missing exon 20, acetylation of alpha-tubulin. And then we found that synergistic effect of 2 drugs have a much higher effect than each one of them alone. I was really blessed with outstanding students. Many of them are currently group leaders in different institutes in Israel. I'm sure that many others will be. And the people that are involved in this project are Shiran Naftelberg, that generated the conditional knockout mice and did most of the collaboration with Eran Perelson, Maya Donio, that found the signal transduction pathway, Sivan Yanai, that is doing the nasal admission and the combinatorial effect, Ron Buchner, that generated the humanized FD mice, and Adas Keren, that found the effect of phosphatidylserine on the ICAP gene. This is our collaborations. There are many other stories in the lab that will have to wait not to the last minutes of the last talk in such a meeting. Thank you all for your attention. Have a nice evening.
Thank you, Gil, for a really exciting and impressive talk. Questions? Well, so if no more questions, I would like to thank all the speakers of this last session. Oh, sorry.
When you do the combination of drugs, you're actually affecting the property in every cell. You try to do it by TSA. What about 5-ASA-C, for example? Does it give the same effect?
Okay.
How low you can go with the dose of TSA to get an effect?
Okay, so what Yossi basically is saying is that treatment with TSA is going to be toxic. Also 5-ASA. 5-ASA is a reagent that removes DNA methylation, and we've demonstrated that DNA methylation also affects alternative splicing. So 5-ASA is something that we are doing right now, but what we are doing, TSA is really toxic, but there are other HDAC inhibitor like valproic acid that we are testing currently, and valproic acid has been approved for other diseases to be used, and it's quite commonly used with relatively minor side effects. So this is something that we are testing right now.
So, on behalf of all of us who remained here and those who have really missed an exciting session and are not here, I would like to thank Miguel, Udi, and all the other people who were involved in making up this great meeting, which I am sure will instill a lot of collaboration, not only between scientists in Israel, but also between Israel and between science in the academia and the pharma. And I'm looking forward already to the next one, next year. Have a nice evening.